My name is Klaus, and I've been working together with Henrik for the last, I don't know, eternity, more or less, 30 years at least. I should talk to you about genetic causes of high, and then I should tell a little about the the possibilities within genetics. And really, what I'll be talking about, it's a lot of methods, so it will be maybe a little difficult for some of you to follow, but shout out if I need to repeat something. So back to basics, really. As you're not molecular biologist or geneticist, I'll have to tell you a little about how molecular biology really looks at things. And what we see is that that DNA is always translated into RNA, which is translated into protein. And the protein is really the machinery of the cell that makes things work. But when we analyze the genome, our DNA mass, we are really only looking into approximately 1% of everything. So there's a lot of things that we don't really analyze. And then again, the DNA is our blueprint. It's the basis of everything that constitute our lives, and codes for the proteins that are the machinery. But during this process, you have RNAs that are translated into proteins. You have RNAs that regulate DNA and proteins, and all that is becoming pretty complex, that you have a lot of RNAs involved. They do protein synthesis. They are modifying other RNAs as enzymes. They are regulating DNA, RNA transcription and translation. And then in the end, actually 10% of all our RNA is viral. It comes from viruses that are just endogenous, jumping around, making a mess out of our genome. But actually 80% is translated in some way into RNA molecules. So you have we are only looking at 1%, but really 80% are in work, in place in our bodies. So you have this tiny molecule, the DNA, constituted by 4 bases that encodes all our proteins. And if you look into the complexity, you have 3 billion bases from mother, 3 billion bases from father. It's just to understand how much data you're really looking into. If you consider that you have the DNA is consisted of 4 bases, A, C, G, and T, and 3 bases constitute the code for an amino acid called a codon. If you consider that as a word, and you consider a normal book page of 840 words per page, then it's equal to 2 million pages of reading, or 50 years of reading if considering a normal individual. So it's a lot of data to look at. Another way of looking at that, it's that you have 1.8 meter of DNA per cell, which means that in a normal beef of 100 grams, depending on your hunger, you have 12 billion cells, which actually equals 22 million kilometers of DNA. Or another way of looking at that, 30 times back and forth to the moon in one beef. So it's a lot of data, and it's what you really have to look into. You have this DNA, the double helix. It's inside our chromosomes, constitutes our chromosome from one end to another. It is one DNA strand. This DNA is packaged inside a cell, inside the nucleus of the cell. Okay, so it was the hard part of molecular biology. CHI is a heterogeneous disease, and as Monica told you, some of the most important genes is the ABCC8 and KJ11, which constitutes our potassium channel. But actually you have more than 30 genes known to be involved in CHI. You have a considerable genetic heterogeneity. You have defects of methylation, you have changes in chromosome numbers, and then you have huge deletions, meaning that you delete several genes or involve several genes at a time. The most common causes are listed here. You have the ATP channel mutations of ABCC8 and KCJ11, which is actually the majority of the patients, 30 to 60%, dependent on population. Then you have those who are have high ammonia levels, which is usually due to GLUT1 mutations. You have those in which you have MODY, diabetes, in the family. Then it's the HNF4 alpha and HNF1 alpha. Or you have those who are exercise-induced, like mutations in the SLC16A1. All these can be analyzed in different ways. And what you can see in the table is that the analysis really only resolves some sorts of mutations. The genome analysis can resolve it all, dependent if you read deep enough in that genome that you are analyzing. Exomes cannot resolve large deletions because you're really reading too low in the analysis. And then targeted panels can more or less resolve all what you want to look at for the known genes. But if you're looking for new genes, you have to go to the genome. Sanger sequence we mainly use if we know the mutation in the family. You have a lot of syndromic forms, and I'll not talk much about it, but so far, as I know, we have 28 different syndromic forms. The most common are Beckett-Wiedemann and Kabuki. You have the genes listed here together with the disorders, and again, you have small deletions, duplications, you have large deletions, you have single nucleotide mutations, and they are really suffering from the same method problems as the common genes of Chi, that you can look for no mutation using Sanger if it's a small mutation, like one nucleotide or two nucleotides missing. But if you have large deletions, you have to use one of the other methods. Then again, sometimes you have really huge deletions that cannot resolve by sequencing. Then you have to use array CGH, as is mentioned in the last pane here. It's a pointer. Okay, and then other disorders, you have methylation problems, like in Beckett-Wiedemann. And so you have to use these methods in those cases. So now we go back to the lab. We go to Sanger sequencing. And this is a method that uses a replication of the DNA target points. You want to analyze a specific part of the DNA, a specific gene. So what you do, it's really you you denature the DNA. It's a double strand. You denature it by heating. Then you add a DNA polymerase, the nucleotides that are necessary for the replication process, and you proceed like that in a cyclic fashion. So what you'll have is you have the DNA strand. You'll add nucleotides, which are the basis of the DNA. You have a deoxy and a dideoxy. This dideoxy is different from the normal deoxy by a hydroxyl group here, and causes a termination in the chain reaction so that you'll have random terminations. And to show an example here, if you want, you have 4 nucleotides, A, C, G, and T. If you want to have a reaction for the T, you add dideoxy T's in 5% of the reaction. And then you'll have random terminations of the replication so that you'll have a termination for each T in the process of generating your replica of the DNA. Over here you have another example. It's adding a dideoxy A, so you'll have random terminations, and in this way you'll generate fragments corresponding to all positions in the DNA that you're analyzing. Analyzing those on a gel by electrophoresis, you'll have the smallest fragment running fastest, the longer fragment running slowest, and you'll generate this ladder so you can actually read. This is the reaction for the C. If you mix it all together, like here the A, G, C, and T, you'll simply be able, based on the run in the gel, to see Engel bases by by and by, and in this way be able to analyze the sequencing. You analyze this running capillary electrophoresis. You simply add the sample, put a lot of electricity, 2000 volt or something, so you keep your hands away really, and then you run it through, and a laser will detect the bases, and you'll get it out on the machine on a computer screen and be able to analyze the sequence. Lately, we have had the possibility of performing next-generation sequencing, and there are basically two forms of next-generation sequencing. One is the proton machine here. The other is the Illumina machine. What you do in the Illumina machine is, again, you have the DNA, you fraction it into small parts, and then you add some what we call adapters. On these adapters, you have indexes, and these are really the main part that you should focus on, because these indexes, they are person-specific, allowing us to analyze many patients at the same time in a single run on the machine, because we can identify the individual patient based on the index that are added for that patient. So this is really the process. You have a small glass slide. It's a double-layered glass slide, and you make the sample run between the two glass slides. This is Lena who is doing that. And what we have is the DNA from the patient that are bound to the glass slide, and we run through some chemical processes, and in the end, we'll have a multiplication of the single DNA strands on this glass slide, and it will be multiplied by 1,000,000 times. So you'll have some small clusters, what we call it. And here you'll have clusters from one patient, here from another, and a third and a fourth. But in the end, you'll have clusters from all parts of the DNA that you want to analyze. And because of these indexes that I was talking about, we can actually, when we do the bioinformatics later, identify the single individual. We perform the sequencing, adding the bases of the DNA one at a time to the sequence, and analyzing for each base added to the sequence in single steps. So running a number of cycles, usually for this Illumina machine, we run 150 cycles, so you'll receive 150 bases of sequence. Here is an example of that. Here you have sequences that have been analyzed for one strand of the DNA as blue and the other strand of DNA as green. Down here you have a live gene, and the blue blocks represent some of these 1% of the genome that we are actually analyzing, the active protein coding parts, and you can see they have been read a lot of times, actually more than 1,000 times. So it's a very good coverage of the analyzer, so you get very precise data. Here's an example from a hyperinsulinism patient. This is a part of the SUR1 gene, and the mutation is an arginine that has been changed into a termination mutation, which means that the gene will simply be terminated, the protein will not function, and this is definitely a pathogenic variation. And what is main from this cartoon is that each mutation that you find, each gene is gene specific and tell you something about the treatment that is optimal for the specific patient. So just as an example of the complexity, we received two small boys from Saudi Arabia. They were cousins. They had Kai, and they had deafness at the same time, which was a little astonishing, as usually deafness is not correlated in any way to hyperinsulinism. But we analyzed for hyperinsulinism, it's what we could do, and we found mutations in the ABCC8 gene. This is a presentation of part of chromosome 11, and we could see that this part of ABCC8 was not present in the sample. But it explained the Kai, but not the deafness. But then we looked into this genome presentation, and I could see this gene out here, USH1C, which is a gene related to a syndrome called Usher's disease, which is a deafness, blindness disorder. And what we found was that the this part of which H1C was missing. So actually, in the end, we could show that the entire area of the genome encompassing both UH1C and ABCC8 was missing, as shown in this Sanger sequence, that the two genes now are completely mixed up, and in the end you have a non-functional gene. This is a machine that I just received in the lab, and it will allow us to perform what we call long reads, reading entire regions like this in one read. Thus you can easily see large deletions, duplications, and even mutilations using this machine, which you couldn't do by any of the other machines that are presented before. Yeah, it's done by Firmpack Bio. So a little about the future, how I see it. Monica before told you about this HK1 glucokinase mutations. These we have recently shown can be present only in single cells or single pancreatic islets. And to analyze for what's happening inside a single cell, you can isolate single cells from, for instance, a tissue or from a blood sample using this small machine here in a microfluidic device. Then the single cell you can isolate into single droplets of lipids. And after that, you can perform full RNA transcription, translate the RNA in the cell, and then you can perform sequencing, and thus generate a picture of the RNAs in which to say the active genes in that particular cell, to say what kind of cell is this? Is it pathogenic? Is it not pathogenic? Is it working normally or not? Here's an example. I'm a molecular biologist. I'm not a clinician, so I'm working in different disorders. This is from a patient who had prostate cancer performing the same analysis. And what we can show is that we can actually identify from urine samples special prostate cancer cells, even they are dead in the urine. We can identify them, and we can show that we can find cancer genes in these cells. So we can identify specific cells from the urine. Same way we could do from blood or any other non-intervening sample. This is an example. If you look into the genetic pathways that are related to the RNA expression, it means what is happening inside that cells, what RNA are producing, what proteins, what kind of regulation do you have in the cell. Here, this is an example from diabetes. Here you have diabetic patients, here you have control patients. So we can definitely differentiate different kind of patients from each other based on the RNA profile of the individual cell. And a little more for the future. We have a number of non-inherited genetic changes, like if mutations are happening inside the pancreatic islet itself, we can isolate plasma cell-free nucleic acids. It means DNA which is not coming from the cells of the blood, but actually from floating free within the plasma. These DNA, RNA that are floating in the plasma are usually residing in something that we call exosomes, these kind of things. And we can isolate specific exosomes using cell type specific antibodies to analyze these exosomes. And here's a cartoon of that. For instance, if this was a beta cell, it would release exosomes containing material that are specific for that exosome. For that particular cell, it could be a beta cell. So you would have the RNA, the DNA, the proteins that are specific for that beta cell. These we can isolate using beta cell specific antibodies, and then we could sequence the content of this, and thus get an idea about the situation of the pancreatic beta cells just looking at RNA presented in the plasma. Thus, no surgery, not interfering much in the patient. Another way of going onwards with research in these areas are to use what we call NGS trio exome studies. What you have here, it's an example of that. You analyze, you analyze using sequencing again, as described before, but this time you use the information from both the father and the mother and the child. So if the child is ill, nothing is happening in the parents, then you know it's probably something recessive, and you should look for a recessive gene. If both one of the parents are ill and the child is ill, then you know that it's probably coming from that parent. So in this way you can start looking for new genes. Okay, then a few days ago I was forwarded some slides from Sarah Flanagan, and here you have Julia and Jane, who are responsible for the Open Hyperinsulinism Gene Project, and which is running, as Klaus described very nicely. And here's what has been found so far. More than 100,000 families have been analyzed from 63 families, 63 countries, and here are the main part of the mutations, as described many times now, ABCC8 and KCJL11, which has been found in 523 families, which is 52% of all families analyzed. Klaus, together with Jasmine and Christine, is doing some work together on HK1, and they have been analyzing and found mutation in so far 89, and of these they have found the mutation present in 31 of the patients' families. And here Joanna has been looking into the presentation of hyperinsulinism throughout life and found that the vast majority is neonatal or infancy. You have some few, 15% in childhood and very few in adulthood, but vast majority it's very early. And then you have some discrepancy in treatment response. You have 63 diazoxide treated, 70% is with both octoroide and diazoxide, and then 2% is octoroide treated. It was just the slides I received from Flanagan a few days ago. Okay, thank you very much.